Okay, hello everyone, and welcome to the Psychiatric Genomics Consortium Worldwide Lab Meeting. I'm Cathryn Lewis, I'm one of the PGC's Executive Directors, and I'm Professor of Statistical Genetics at King's College London. So the Worldwide Lab Meeting is our regular forum for presentation of PGC-related research, with the idea of generating discussion and providing training for anyone who's listening. And so a very warm welcome to you all, whether you're listening live with us today or through the recording on the PGC Genomics YouTube channel. These sessions are open to all. Please do forward the information to your colleagues and collaborators who might want to join us. Today's session is on the latest advances in the field of fine mapping, really important area as we move our GWAS forward. And we've got three outstanding speakers in the field who will cover some of the progress and also the challenges involved in this step, moving from association to causality. So we have three speakers, Johnny Coleman from King's College London, who's going to talk about fine mapping major depressive disorder using sample level linkage disequilibrium, and Maria Koromina from the Icahn School of Medicine at Mount Sinai, who will talk on integrating multi-omics and multi-ancestry data for fine mapping in bipolar disorder, and Jennifer Asimit from the MRC Biostatistics Unit at the University of Cambridge on how diverse ancestry data can improve our fine mapping strategies. And just two housekeeping points before we move on to the seminar. We're going to run through all the talks first, and then we'll keep our discussion session to the end so we can cover all the talks together. But if you've got a burning question, please do put it in the Q&A link there so you can post it while you remember, and then we'll come back to it at the end of the session. And you can post anonymously if you prefer, and you can do that at any point during the talks. So to start off, I'm going to ask Maria Koromina from the Icahn School of Medicine to start with her talk on bipolar disorder. Are you okay to share your slides, Maria?
Yep. Can you all hear me and see the slides? Oops.
We can hear you and see the slides.
Yep.
We've still got...
Perfect.
Brilliant. Right.
Okay.
Over to you. Thank you, Maria.
Thanks, Cathryn. Thanks for the kind introduction as well. And over the next couple of minutes, I'll just go through different fine mapping strategies that we can use to refine bipolar disorder risk genes. So first things first, as Cathryn mentioned, GWAS studies are super helpful, but there are still things that we need to resolve. They can help us identify specific regions of interest, which are associated with specific traits. But as you can see, because of the LD, there is a lot of noise. So we need to apply fine mapping Bayesian methods to get specific credible sets. These credible sets, as you can see here, can contain the likely causal SNPs. So we're more confident that one of these could be the likely causal or causal ones. There are, though, factors that can influence fine mapping. One of them is the local LD structure. Also, the sample size of our GWAS, we ideally need more than 10,000 samples, the number of causal SNPs in a specific region, the LD reference, which Johnny Coleman is going to touch base also on that, and whether the causal variants are actually measured in the specific region. Another question that we often get asked when we do fine mapping is that why don't we take simply the top associated SNP? And in the scenario where we have one single causal variant, as you can see here, that should be quite straightforward. But in the scenario, which is the most usual one, where we have more than one causal variants, we actually need to apply fine mapping to understand which one is the causal one and get, as you can see here, the credible set as well. So what we've done with the third phase of the bipolar disorder GWAS, which, this paper has now been accepted for publication in Neuroscience, is that we began with the 63 genome-wide significant risk loci. And just for now, we just excluded the MHC locus because of the complex LD. And we applied condition analysis to get down to the number of independent signals in its specific region. And then we applied fine mapping to get the set of likely causal SNPs by exploring different options. So we applied two statistical fine mapping methods, SUSY and FineMap, and two functional ones, polyfin SUSY and polyfin FineMap. And we also explored different options for the LD, such as the HRC, UK Biobank, ensemble LD estimates, and we also excluded LD. And all of these steps, which I just explained, are covered in a pipeline called Safari, where you can run it in a scalable way and get these results. And we still, though, needed to map these likely causal SNPs to genes. And to do that, we also explored different options, one being classic variant annotations, also checking for a physical overlap with Plexic data, like interesting, epigenomic data. We applied SMR using brain bulk data, EQTLs, SQTLs, methylation QTLs. And we actually ran SMR with probes in the cis region with FineMap SNPs as the QTL instruments with that tweak in the SMR. We checked for an overlap with rare variants, and we also tried to assess whether the causal effect sizes from the fine mapping can affect polygenic risk scores. But because of the sake of time, I'll just focus on the area, in this purple shape. So after applying all these different methods, as we discussed, we tried to assess, the number of causal variants above specific posterior inclusion probability thresholds. This means that actually, the SNP could be causal. That's what this probability is measuring. And from what you can see here, we identified quite a few SNPs with high probabilities of being causal. And these numbers were a little bit lower, for SNPs with a threshold of 0.95. The other thing we tried to assess, sorry for that, is actually, the heat maps with the Jaccard indices. So we tried to assess for SNP concordance and, see whether specific methods and approaches actually harbor more of the SNPs. So as you can see here, sorry, I don't know why these animations are like that. So as you can see here, when we compare statistical fine mapping methods, even with different LD approaches, we got a higher level of concordance in the results. But when we actually compared statistical versus functional fine mapping methods, even within the same LD panel, this similarity, actually decreased. We have a lesser degree of concordance. As I mentioned before, all these different methods and all these different approaches give us sets of likely causal SNPs, but we aim to get a union consensus set. This means we kept SNPs if they were found in at least 2 of the 4 fine mapping methods within each specific LD panel. And I'm just going to highlight, a couple of interesting ones in this plot, where you can see also the shape and the size of the different dots or circles encoding for the posterior inclusion probability. So some interesting ones are found in the sodium channel. We have 2 specific consensus SNPs in this region. Trunk 1, which is, I think, a known candidate. KRC7A, which affects lithium response. Furin, another specific SNP in a certain intergenic region, which is also found from all these different approaches. And another SNP, in the THRA region, ERBB2. But as I mentioned before, this is nice, these are specific SNPs that we are actually, quite interested in. We still need to map the SNPs to the genes. And in order to actually do that, as I mentioned before, we applied different approaches. One of them is the physical overlap with Plexic data. And if the specific SNP, which you can actually see here, this union consensus SNPs, and then all the different line of evidence. So if the SNP was located in the promoters, we actually applied the corresponding gene name. And if the SNP was found within specific active enhancers, we use this Plexic data to try to map, the SNP to the gene which is actually affecting. And we're actually denoting these genes, within these shapes in the tile plot. The other thing we did, as mentioned earlier, is apply SMR with the FINOM SNPs as the QTL instruments. And we did that for different types of QTLs from the brain bulk tissue, eQTLs, splicing QTLs, methylation QTLs. And from what you can see, this approach revealed causal relationships between these union consensus SNPs and bipolar disorder through changes, in the gene expression, as you can see here, or the splicing or methylation of interesting genes. And as I mentioned before, we also applied variant annotations, and you can see two specific genes that harbor missense variants. So from the QTL from the SMR approach as well, some of these interesting genes are the sodium channel, TRANK1, the calcium one, furin, which is a known candidate, and FES as well. And I want to just highlight this example here. I think it nicely illustrates what fine mapping can resolve for us. So we have the sodium channel gene, as I mentioned, and we have two specific SNPs, which I'm highlighting in the union consensus set, either identified from statistical or functional fine mapping. And you can see one of them falls within promoter regions in neurons. And we have this interesting results from SMR, which I highlighted before. And of course, sodium channel is involved in the neuroscience. This sodium channel gene is involved in the neuroscience of sodium channels and implicated in other disorders such as epilepsy. So some key take-home messages before I move on to the next session of the talk is that through this comprehensive approach, we identified 17 high-confidence causal SNPs, in 63 bipolar disorder risk loci, and these SNPs, these consensus ones, were mapped to 22 candidate genes, highlighting a few here as well, which we further supported with epigenomic data, QTL data, and rare variant evidence data. And you can run the fine mapping part through Safari, which is an open source and scalable pipeline. But you can ask, and I will ask as well, can we also implement multi-ancestry fine mapping methods to actually further improve the fine mapping resolution? And with that, we actually mean whether we can leverage the differences in the LD patterns between the different genetic ancestries to identify common causal variants. Because the previous part and whatever I discussed has been done, predominantly in European data. So in the following project, which I'm going to explain, and it's actually my NARSAD award from last year, so in collaboration with Panos Roussos and Ole Andreasen. So what we aim to do is actually build a multi-ancestry fine mapping framework. We can begin, and we will begin from this 298 genome-wide significant risk loci from the latest freeze of bipolar disorder, and apply again fine mapping approaches, but this time using SUSY-X, where you can input your ancestry-specific summary statistics and ancestry-matched LD matrices, get a set of likely causal SNPs, and then similarly as before, try to map them to specific high-confidence genes through SMR, which is one of the approaches that we will implement, and use brain bulk eQTLs, SQTLs, and methylation QTLs, and single nuclei eQTLs as well for the cell type resolution. And of course, can still use Plexic data for additional annotations. Again, for the sake of time, I'll just focus on the first part with the SMR approach. And just briefly, I want to highlight that we will plan to actually further translate these results by estimating functional PRS, similar as before, try to see if the fine mapping effect sizes can actually affect PRS accuracy and portability, and identify high confidence genes as well, which are actually affected and targeted by drug compounds. So what we're showing here is that we use these 298 genome-wide significant risk loci to begin with, and we actually try to compare SUSY-X when we include just two populations, let's say European and East Asians, again, from the latest freeze of bipolar disorder data, and also run SUSY-X with 4 populations: European, East Asian, and the admixed ones, Latino and African American. And these results here, we compare them with what has already been done, where we use pretty much the multi-ancestry meta-analysis and run Polyf and SUSY. So the main take-home message from that, as you can see, is that the mean posterior inclusion probability is actually improved when we include more non-European ancestry data in the fine mapping. And this calls, for actually using such methods to improve the fine mapping resolution in our data. And as done before, we actually followed the similar approach. We performed SMR using these SUSY X SNPs, these likely causal SNPs, as our instruments. And what we're showing here is that we can identify specific genes with multiple lines of evidence. They can either have, some SNPs, with high posterior inclusion probabilities. From SUSY X, we're also showing which one is the closest annotated gene, and also which genes we pick up when we use brain bulk QTLs and brain single nuclei QTLs. And I want to highlight this example here. So one of them with RGPD8 gene. So what we saw is that there is a causal relationship between this likely causal SNP and bipolar disorder through changes in RGPD8 gene's expression in neurons, excitatory, inhibitory, and astrocytes, and also the bulk tissue. And I don't know if you can see, we also have quite a few signals from methylation. This means that potentially methylation changes within the annotated genes can be associated with bipolar disorder risk. I think this is possibly the question I get asked most, like, it seems that we're just, listing genes from different approaches and different QTL data sets. And usually, by the end of most talks, we're actually, okay, which one or which ones are of interest for people and which ones can they follow up? So it kind of seems, that people, including me as well, we can be lost when we do that. So I'll just try to summarize my talk with 2 different points. So the first one is whether the GWAS gene can actually differ from the EQTL gene. It's just important to keep in mind that QTLs map variants to molecular traits, which is the expression, but GWAS actually maps specific SNPs to complex phenotypes through LD and through changes in LD. And QTLs can show larger cis effects on single genes. GWAS hits usually have smaller effects across more broad loci. QTLs also can be tissue, cell type, condition specific. GWAS signals are more aggregated with limited cell type resolution. So it's kind of important to keep these things in mind when we do this work. And just listing a few challenges, but I'm pretty sure the next few speakers will do the same. And, we can have this discussion at the end. First one is which multi-ancestry method is optimal for fine mapping. And I'm saying multi-ancestry because we're moving, in this era, where we add more and more diverse data. So should we focus on the multi-ancestry meta-analysis compared to the ancestral GWAS summary statistics? Or should we fine map the first, or should we have methods where we just add in the ancestral GWAS summary statistics? Can we efficiently resolve regions with complex LD, and if yes, how can we do that? What's the best way of integrating multi-ancestry omics data with multi-ancestry GWAS as we proceed? And any differences, in ancestral proportions of either data sets, can they actually affect causal inference? And of course, it would be great and ideal to actually increase the sample sizes for single cell EQTLs and single nuclei EQTL datasets. Just finish here. Hope I didn't take too much time from the next speakers. I'd like to thank the Mullins Lab, Neve, for all her guidance and support in this work. Ole and Kevin from the Bipolar Disorder Working Group, everyone from the Mullins Lab, all the people I'm listing here, and our funding. And I'll just stop here so I don't take more time from the next few speakers.
Thank you very much, Maria. That was a fantastic introduction and a really clear description of on fine mapping and your results in bipolar. If people have questions, please do put them in the Q&A and we'll pick up with those later. And then I'm going to hand straight over to Johnny, who's going to talk about fine mapping using sample level LD in major depressive disorder. Perfect. We can.
Thank you very much, Cathryn. I think you can see my slides. Can you hear my voice? Yes.
We can.
Wonderful, fantastic. And thank you very much, Maria, for covering my obvious lateness. So yes, as Cathryn said, I'm going to talk today about some of the things we've learned from our efforts to fine map major depressive disorder over the last few years. I'm going to briefly talk about single variant fine mapping, which was a component part of the main PGC paper that was published earlier this year. But I'm primarily going to talk about our experiences within multivariant fine mapping, and particularly the role of sample level LD. So as Maria will have introduced, the idea of fine mapping from a Bayesian perspective is to model causal variants, or one or more causal variants, and model the effect of those variants on the local variants around them, whether that be scaled by the linkage disequilibrium or by the correlation between their effect sizes. And then we're fitting how well the observed data matches those different models to identify which is the putative causal variant. So in this case, this data would best fit this model with the red variant indicated there. So given that basis, we're then looking at a relationship between single and multiple variant analyses. The benefit of single, or perhaps one assumption that single variant analysis holds, the multiple variant analysis doesn't hold, is this idea of no allelic heterogeneity. So this idea that there's a single causal variant that's causing the association we see. And that may be biologically plausible or implausible depending on the different loci. It makes sense that if a variant is affecting a specific gene, that there could be multiple ways that that gene is affected and a convergence of multiple signals onto a single biological pathway. So there is a reason to kind of doubt that hypothesis. But the benefit of single variant fine mapping is that mathematically it doesn't require estimates of linkage disequilibrium between the variants. Something to bear in mind, both in terms of single variant fine mapping and in terms of multiple variant fine mapping, is both that what we're doing is not truly looking for the causal variant. We're looking for, of the variants that we're modeling within the fine mapping, which is the most likely to be causal, which best fits the role as the causal variant. And if we don't actually have the true causal variant within our set, then we're going to see false positives and we'll get reduced power. And another way that we get reduced power and another way we get false positives is also between cohort heterogeneity. So many fine mapping methods have been developed based on large single cohorts. And when you move into multiple cohort analyses, you start to generate these biases and these errors that affect the way that fine mapping works. And there's a particularly good paper by Sinai et al. Describing that. So to launch into depression, to launch into some work, I'm going to begin with this data set, the European ancestry subset of PGC MDD3. We have 500,000 cases, 3 million controls. As I said, I'm focusing on European ancestries. We actually ran this same pipeline for the diverse ancestries data. For time, I'm not going to talk about that. Also, we ran this exact pipeline in the diverse ancestries data. I'll highlight some places where that doesn't quite work as I go on. So we were analyzing within each of the 564 loci that we're looking at, and I'm going to focus on variants with putative causality, so above a posterior inclusion probability of 95%, and the credible causal sets that are comprised from that as well. Here we're focusing specifically on functionally informed fine mapping. So this is the PolyFun plus SUSY pipeline that Maria will just have talked about. is built on stratified LD score regression and weights the priors of the variants going into the fine mapping by the enrichment of heritability that their epigenomic characteristics show in the whole cohort. So leaving aside the chromosomes that the variant is held on, so odd chromosomes versus even chromosomes, Polyphon uses the whole summary statistics for the given trait to generate priors based on functional enrichments in the data itself. And then it passes those priors to Susie that does the actual fine mapping to identify variants. So in this aspect of the work, we focused on single causal variants, specifically within the windows defined by linkage disequilibrium clumping for each loci. And obviously the benefit of this is that we don't need LD estimates for the fine mapping itself. However, because we're doing functionally informed fine mapping, this relies on LDSC, so it relies on the reference linkage disequilibrium that's part of stratified LDSC, which is one limitation of then applying this to a diverse ancestry data set, is you don't have the appropriately matched population level LD. Here we've defined credible causal sets as simply the ranked list of variants by their posterior inclusion probability, with the credible causal set being the cumulative number of variants it takes to have a cumulative posterior inclusion probability above 95%. So to cut to the chase, we identified 24 out of 564 loci that could be explained by a single causal variant with a posterior inclusion probability above 0.95. I'll leave this slide up for a couple of seconds for people to identify their favorite gene. One thing to note is that in all cases, the p-value for the variants are less than 5 times 10 to the minus 8. That's not something that we selected. It's just something that falls out of the model. It probably reflects that we are really only at the beginnings of power for what we can do with fine mapping here, that we're really only finding the variants that are sufficiently well powered that they are genome-wide significant in the study as well. Many, if not all of these, will also be the lead variant in the locus as well, which probably again reflects power. The same for the second half of the 24. I'd highlight that variant on chromosome 15, the furin variant, which the eagle-eyed of you will notice also came up in Maria's talk on bipolar disorder as well. And then looking down into the credible causal sets, we can identify a number of relatively small causal sets, 224, where the total number of variants that it takes to get to a cumulative posterior inclusion probability of 95 is less than 10 variants. So we've got quite a large number of loci where we can narrow down to a relatively small number of variants for further examination. And we can also look at the extent to which the entire causal set lies entirely within one gene. And in that case, that was 234 loci where that was the case. So we get some insights from single variant fine mapping, but this comes with the assumption of allelic heterogeneity. So we're assuming that there must be one causal variant. We can relax that assumption by moving to multivariant fine mapping, but in order to do that, we then need estimates of linkage disequilibrium. And that's where I want to spend the majority of this talk. So this was what we did in my recent work funded by the Medical Research Foundation in the UK. And most of this work was done by this gentleman on the screen, Dr. John Vincent, who was my postdoc doing this work. So a major challenge we face in MDD in particular is that much of the data that goes into our GWAS is provided only at a summary level. We don't have the individual level data for a lot of our work, and this is in contrast to, for example, bipolar disorder, where a large amount of the cohorts do give us individual level data. So what we set out to do was gather together, summarize linkage disequilibrium information, both from the cohorts which we had the individual level data for, and also from cohorts provided by collaborators, and then conduct fine mapping using both that in-sample LD and the haplotype reference consortium LD to compare how that performed. So this is the overall kind of pathway, we have that same set of GWAS summary statistics, so the European subset of the PGC3 work. And we've gathered together LD matrices that reflect around 600,000 SNPs of interest across those 564 loci. We meta-analyzed LD matrices, and I'll talk about that in a little bit more detail in just a second. And then we performed fine mapping analyses using that same SUSY plus polyfun pipeline. And we also integrated FineMap, which I'll talk about in just a second. So to briefly discuss LD meta-analysis, we had 45 cohorts available to us with individual level data on Snellius, totaling about 64,000 individuals. And we then had summary LD matrices that 20 collaborating groups were able to provide us with just under 1.9 million individuals. Unfortunately, there were 5 cohorts for which we couldn't gather data. These were mostly pharma cohorts where data access is not so straightforward, and most notably 23andMe also we weren't able to secure data from. But the data which we did have, so that's about 50% of the total sample size of the analysis, we meta-analyzed together, and we meta-analyzed those weighted by sample size. So we have each of the pairwise LD estimates, and they're combined weighting by sample. So here is our broad analysis overview. There's quite a lot going on here, but hopefully the color scheme will eventually make sense. So we have summary statistics going in, both from the full PGC MDD3 analysis and also from the subset for whom we have our matched LD panel. And then we have that matched in sample LD panel and the external haplotype reference consortium LD panel. So we first put this through polyfun plus the FineMap pipeline in order to predict the number of causal variants in each region. So FineMap is able to produce an estimate of this. And we then pass that into 3 sets of analyses. So first, our core analysis, where we're matching the priors from the 65 cohorts for whom we had LD data with their own panel. A secondary analysis where we took that same summary statistics and matched it to the external panel. And what we're calling an extended analysis, where we took the full PGC summary statistics and used our in-sample LD panel as well. And all of this was run as polyfun plus SUSY using the K that we get from the FineMap aspect. So within that, we're then looking much in the same way as we did before within each window, but now we're looking for multiple causal variants, and we can define credible causal sets in the way that SUSY usually does, which is not just the sum of variants where their cumulative PIP adds up to greater than 0.95, but also that the minimum R² between variants must be no smaller than 0.5, i.e. that set of causal variants truly hangs together as a credible causal set. It's not that you've got some outlier that isn't actually correlated to the rest that happens to just lift your cumulative PIP above 0.95. So the questions we wanted to ask were, are we able to identify multivariant credible sets with high confidence when we're using sample accurate LD? And is that also there when we look at population accurate LD? So in other words, do we actually need this rather torturous process of gathering sample accurate LD, or can we get away with population accurate LD in this case? We then wanted to see, are our results consistent when we look at the core analysis and when we extend to the full PGC3 results? And then if that's the case, can we then identify multivariant credible sets in that full PGC3 analysis, where we obviously have more power because we've got the full sample to look at? So a first hurdle that we discovered was that, much in the same way as fine mapping in general seems to struggle with between cohort heterogeneity, FineMap also seems to struggle with meta-analyzed data, and especially when you're using external LD. So what we found was we ran the analysis multiple times, and we found that we both got lots of regions where the estimate of the number of true causal signals ended up with the maximum value, which in our case was 5, which essentially means that FineMap just isn't able to make a decision on how many causal variants there truly are. But we also found a lot of regions where our estimate of causal variance was unstable across multiple runs. So we didn't consistently get the same K when we ran FineMap multiple times. And this makes sense. FineMap is a stochastic algorithm, but it's not, you can't really put a lot of confidence in a region where you can't get a consistent estimate of K. And you can see it varies a little bit between the different analyses. So we actually got our best analysis with our best results with the HRC panel, but when we go to the external analysis, when we're adding in probably most notably the 23andMe data into the summary statistics, we find the stability of our regions drops considerably, and of 564 regions, we're only able to get stable estimates in 275 of them. This doesn't seem to be obviously driven by specific regions. It's not obviously a factor of specific LD panels or some stats, and the overlap between these regions isn't particularly high. So looking between the core analysis and the secondary analysis, or the core analysis and the external analysis, we typically don't, it's not typically the case that both of the regions are unstable, or sorry, the regions in the same region in the two analyses is unstable in both cases, or that it falls to maximal in both cases. It seems to be as often the case that it's stable in one and unstable in another as it is stable in both, unstable in both. So leaving that point on instability aside and focusing only on stable regions, what can we then do in exploring multivariant credible sets? So our first finding is that actually a single variant is sufficient in the majority of loci, and this is also consistent with, for example, what the previous schizophrenia analysis found in their major paper as well, and I think also consistent with what was found in bipolar disorder as well. And that again might be a factor of power, as well as a biologically real finding that there isn't actually true allelic heterogeneity in many of these regions. So we took forward 47 regions, of which 34 of them had estimated causal signals of 2, and 10 and 3 for 3 and 4 signals accordingly. So within those 47 loci, we identify 40 credible sets, including 12 loci where there are multiple credible sets, and 3 loci where we not only have multiple credible sets, but those multiple credible sets are comprised of relatively few variants. So just to give a couple of examples of what that looks like, this is a locus near the gene CIR1, and you can see on the left, ringed in red, you have a credible set that lies directly over CIR1, and on the right, a set that lies a little bit further down and perhaps implicates other genes. However, in some cases, your multiple credible sets are actually a bit illusory, and we have, for example, here a set of variants that lies over a particular region in PCDH9, but actually is realistically probably better described as a single causal region with a couple of variants that, for whatever reason, don't fit the LD properly. Perhaps they're not genotyped in as many cohorts, or they have different effects. And similarly, this is that single variant I highlighted earlier near Fesfurin, which in our results actually comes up as a two causal variant set. And actually, when you look at the data, it kind of seems like a second causal set is just shoved in rather than being kind of a reality. These two variants, whilst they have very high posterior inclusion probabilities, have basically no p-value of being associated with depression. So this again is not really a true multi-causal set. It's really the model trying to find a second set because it's been told that there is one. So for our second question of whether these results are consistent, largely to kind of skip to the punchline, what we find is that when we focus down onto the stable regions, we tend to get the same estimate of causal variance. That seems to be very consistent, and that generally we see pretty much the same credible causal sets. They're at least overlapping. The ranking of the SNPs sometimes is very high, and the correlation of the posterior inclusion probabilities across SNPs that are in both credible causal sets is very high. And that's shown here as just the direct correlation. Even when we have off-diagonal effects, they tend to be relatively small in size. For the purposes of time, I'm just going to skip a further statement and just skip to that same comparison between the core analysis and the external analysis. So this is using the same panel, but looking at just the GWAS that we see, just the GWAS of that data and the GWAS of the full data. And what we see there is much less consistency. So one of the kind of key takeaways we find is that actually it really does seem to matter that you lack the true underlying LD in this instance, and so we have to be quite cautious about the conclusions that we make from this analysis. There is evidence of consistent variance. But there is also plenty of places where our estimates do not agree between those two things. And that's challenging to unpick because we've got a mixture of power and accuracy of LD going on in that. So to end with conclusions, what we find is one of our main conclusions, and something that needs to be borne in mind for future research, is that FindMap is unstable when you're assessing multi-analyzed data and you need to repeat it to determine robustness. MDD, like many other disorders, seems to be primarily composed of single signal loci, but there are multiple signal loci to find. If we seem to find reasonably good matching between population matched LD and sample specific LD, but you always kind of want to go and validate things. And losing LD, not having the LD of a sizable proportion of the sample, does seem to create instability, and it's difficult to judge to what extent that's a power thing and to what extent that's an absence of LD. But there are still some interesting loci that we think are worthy of exploration. So with that, I will thank John Vincent, who did a large amount of the work, and thank the various funders of this work as well as others who contributed to this. And thank you for listening.
Thank you very much, Johnny. Super talk and all the details on the MD analysis data. Reminder, you can put questions into the Q&A, and we'll move on to our third talk, which is from Jen Asimit from the MRC Biostatistics Unit in Cambridge, who's going to move on to talk more about multi-ancestry mapping. Jen, do you want to share your slides? Perfect. We can't see or hear you at the moment, Jen.
There you go. Sorry about that.
Super. Great. Off you go.
Okay, great. Thank you. Okay, I'll just get straight into it because I know we're limited with time. Okay, so I'll briefly go over some approaches for find mapping that have been developed in my group. So first, I'll go briefly over multi-trait find mapping, then look at multi-group or multiple genetic ancestry find mapping, and finally combine the two together so to gain the advantages of both multiple traits and multiple genetic ancestries. So why would we want to jointly find map multiple outcomes? I think it's quite clear intuitively even. So because related diseases or traits likely share some causal variants, by leveraging information between them, we could increase our find mapping resolution. And that's especially if they do share the causal variants. If they don't have shared causal variants, those gains are not going to be seen. And even more importantly is if you have smaller sample sizes, as is common with non-European genetic ancestries, you can actually improve your resolution by sharing information between traits without the extra cost of increasing your sample size. And I'll show a nice example that illustrates that. And by looking at multiple traits together from different diseases or different diseases together, we can get insights in multimorbidity. So here are some of the previous multi-trait find mapping approaches. One of the earliest ones is
Jen, certainly on my screen, we can still see your title slide.
Oh, it's not changing.
No.
Oh, okay. Thanks for telling me that. I'm not sure what to do differently. What did I do wrong?
Ah, that's good.
No?
Yeah.
Okay, let me put it back into full screen. Is it changing?
No,
We've
it's not
still,
the full screen. It's probably showing the wrong screen for you. The full screen maybe opens. But if you just don't do full screen and keep us showing this one and move forward, maybe that would work.
Okay.
we've got features of Flash FM on our screen and now multi-trait fine mapping.
Okay. So shall I just leave it like this then? Or let's see if I can like, how's that?
Yeah,
Changing? Okay, good.
that's
Okay.
changing. Great, thank you.
Great. Okay, thank you. So sorry, there we were. So previous methods with multi-trait fine mapping, one of the earliest ones is fast painter, where they assume you have the same set of causal variants by allowing for different effect sizes between traits. When the causal variants aren't actually shared, though, the precision will not be as good, but does output trait-specific results. And what that means is that for each trait, you're going to get a credible set of potential causal variants for each trait, as well as the posterior probability that that variant has a causal association with each trait. Flash FM is a method developed in my group, and this one allows traits to have different causal variants, and it also allows missing trait measurements for individuals, as does Fast Painter as well, and it can be used in meta-analysis summary statistics, and like Fast Painter, it outputs trait-specific results. A more recent method now is MV Suzy. It's a preprint. It allows traits to have different causal variants like Flash FM, but it does require complete data. So if you have 5 traits and you have individuals have only 4 traits or 3 traits or 2 traits, those individuals that don't have traits measured for all the trait measurements will be excluded, and you have to run your GWAS again in order to use that. And it doesn't output trait-specific results. Instead, it gives a 2-layer output. So instead of saying this trait has a posterior probability of causality for, this variant has posterior probability of causality for this trait and this trait and this trait, it gives posterior probability that it's causal for at least one trait, and you need a second metric to decide which traits are affected by the variant. And a feature of all these methods have, a restriction actually, is that they do require that the variants are measured for all traits. So Flash FM, it's flexible and shared information fine mapping. It is in a flexible framework. So really, I want to stress that it is actually a framework, so you can integrate. It relies on single trait fine mapping as input into it. So it could be integrated with FineMap, JAM, or more recently Finimom. So any of those methods or any other method that does give you a model posterior probability, the posterior probability of some combination of SNPs together. So the shared information aspect, where that comes in is that the prior for your joint model is going to change depending on what variants are in your shared model. So for example, if you have just a simple case of 2 traits and you have a 2-SNP model, these 2 SNPs here, and then trait 2 has completely different SNPs, your prior for your joint model is just going to be basically assuming like independence type kind of prior, just you multiply your priors together in some anchoring term, which is a combinatorial factor. That's not really, don't want to stress that. If they do have a shared causal variant, as in trait 1 and 2 here, where they share this one variant here, the prior is going to be upweighted by this factor kappa, which again, it's a combinatorial measure. What I want to emphasize here is that if indeed the traits actually don't in fact share causal variants, what's going to happen is you get back the same results as you would from single trait fine mapping. So there's no increased false discovery rate. There's no loss from doing this multi-trait fine mapping. This is the example I mentioned earlier. So in one of the largest actually African GWAS is a size 6407 from Uganda. Using JAM single trait fine mapping, the variant that was prioritized was this variant here. And I give all the numbers down here if you like to see the numbers instead of the figure. When we ran multi-trait fine mapping on 4 lipids traits in the same sample, it gave a different answer, as you would like to call it. So it's RS742, which is a well-known variant for LDL cholesterol. And the trait here is HDL. Sorry, I didn't mention that. And the nice thing is that when we run JAM, so going back to single trait fine mapping on a substantially larger sample from a meta-analysis of sub-Saharan Africa, we get an answer that agrees with Flash FM on the much smaller sample. And you can see the numbers down here. So it's just a nice proof of principle of how leveraging information between traits when there are shared causal variants, we can get these nice gains similar to increasing sample size. The other two talks also touch on function annotation. So just very briefly, in the Magic Consortia, we have combined function annotations with FlashFM. And there was a nice decrease in the median credible set size from 58 for annotation agnostic single trait fine mapping using FineMap down to a median of 17.5 if we use FlashFM instead. A nice visual of that. So you can see here a difference. So here's just single trait fine mapping, agnostic, no annotations involved, single trait incorporating annotations, and multi-trait FlashFM incorporating annotations. And you can see the top variant. MPP is marginal posterior probability. It's the same thing as you've seen in other talks of PIP, the probability of inclusion, posterior inclusion probability. It's the same thing. So you can see there's a significant difference between them, that there is actually a trend where you are getting, you can have a better prioritization of your causal variant as you go through these methods. And here you can see the credible set size. So we're going from single credible set sizes, or the yellow ones. So we can go from 2 to 3 to 8 as we go across from single trait, no annotation, to layering FlashFM with the annotation. And likewise, it's a consistent trend across that we're getting heavier with the smaller credible set sizes and fewer of the larger credible sets. Shows that, and there's a preprint here on that work. It was led by Jana Sogson in Ines Barrosa's group in Exeter. Very recently, we published latent factor GWAS FlashFM 0 in Cell Genomics. So this was geared towards where you have many traits. So for example, blood cell traits in the plot here that's shown is for 99 blood cell traits. And we can actually reduce it down to 25 latent factors that we analyze instead. And a nice feature of the latent factors is, one, in terms of interpretation, they will cluster traits that have common underlying biological mechanisms, but also because of the way they're designed statistically, they're uncorrelated, which makes it very easy to scale FlashFM to analyze any number of uncorrelated traits using this approach. And within this, we also include a way, so if you have your observed trait GWAS summary statistics, using those together with some results from your factor analysis, your loading matrix, together with your trait covariance matrix, using all of those together, you can actually estimate your latent factor GWAS, which means you can use this for any other downstream analyses. And the advantage with the latent factor GWAS is that you will find signals that you might have missed using just the marginal trait effects from before. This is a nice example for SMIM1. It's a regulator of red blood cell formation, where I'll just walk you through this very quickly. So each little box here shows a latent factor. It's an arbitrary name. It's ML12, ML14. Those are just the latent factors because through maximum likelihood estimation. But what I want to highlight here is these ones in the blue box are the ones where we do multi-trait latent factor. And you can see we get down to a single variant credible set in each case. The ones in the purple boxes are just latent factor single trait fine mapping, which also gives an improvement, although not as good as the multi-trait. And these other ones are individual, so MCHC, these are individual blood cell traits. And you can see they are in the double digits in terms of their credible set size, though they do all contain the top variant that was highlighted from the other methods. So it just shows the nice gain that we can get by using this type of method. So multi-ancestry fine mapping, you've seen that already. So I think it's well known that you can get nice gains just leveraging the different LD from the different genetic ancestries there. So here's a little cartoon illustration to show that in African populations, you do tend to have the shorter LD blocks, whereas they're longer in maybe European and East Asian. And by combining those across, you can kind of pinpoint much better your causal variant. I think I'm going to skip this for time. I'll go straight. So previous multi-ancestry fine mapping methods, they tend to rely on taking the intersection of the variants that are present in all of your genetic ancestries. But by doing that, you actually exclude quite a bit. So an example is if you look at the APOE gene in 1,000 genomes, there are 928 European-like variants, 1616 African-like, and their overlap is only 690. So if you were to rely on the previous multi-ancestry type methods, you would be excluding 60% of African-like variants and roughly a quarter of the European-like variants, which means those African-specific variants or so on are going to be missed using methods with those kind of methods, which of course at the time were really good for finding those kind of variants, but with advancements, we need to look for alternatives. And so with that in mind, and also wanting to leverage multiple traits together, my group developed MG/FM. So that is taking advantage of improving causal variant prioritization from the similarities in causal variants between traits, and also including the differences in LD patterns across your population groups. And that's the first method to combine both of those features together. And a nice feature as well is that it does include variants that are present in at least one group. So you're able to identify ancestry-specific causal variants as well as shared ancestry causal variants. And when I say shared ancestry, it doesn't have to be shared across the board with all ancestries there. It's not one extreme or the other. It just means like any subset of ancestry shared between them. So you can find anything amongst those. And more recently is SUSY-X, which Maria mentioned as well. It also has the feature where it includes variants that are present in at least one group, but it's a single trait multi-ancestry method. Here, just some simulation studies. Just what we have here is that trait 1 has two causal variants, A and D, just labeled them that way. Trait 2, A and C, so they have one shared causal variant is the key thing here. These are just sample sizes, so equal sample sizes between Europeans and Africans, whereas here's more realistic one where we have 90,000 Europeans compared to, say, 10,000 genetic ancestry African. And when you have a shared causal variant, so the dark blue here, we're just doing single ancestry European /FM. Here's single ancestry African /FM, and because of the tighter LD block, that's why we have that improvement between the two /FMs. MG/FFM is just a single trait multi-ancestry version, so analogous to the framework in MG/FM, which is the yellow one. So there's a nice gradient here where you can see we do get those gains from multi-trait and multi-ancestry, and even more so when we have larger sample size in one of the samples. And in the case if there's no shared causal variance at all between them, then we're not going to gain by leveraging information between the traits. So MG/FM and MG/FM actually have very similar performance because there's no sharing to leverage between them. And I think I'm going to skip that because we are getting close to time. Here's a nice application with the Global Lipids Genetic Consortium, where we applied MG/FM, and you can see here APOB, which is a drug target for cardiovascular disease and hypercholesterolemia, And here we focus on the 4 lipids traits, and you can see here all the variants that were highlighted. So these are the ones that were found by MG/FM, and it all means it was in all five genetic ancestries. Or here it just specifies it was specific to European, Hispanic, and South Asian, or this one was only present in East Asians, and we were able to find those. In the GLGC analysis, they restricted it to a single causal variant, so you weren't able to find those multiple causal variant models. And just showing here where it was found also by UK Biobank, where they used FindMap to do their analysis, so they did allow multiple causal variants. So that's where we're able to find those ones as well. This one, because it was specific to European, Hispanic, and South Asian, and the other ones were prioritized by European. It just wasn't picked up by GLGC, but it was by UK Biobank, and so on. I won't go through all the details there, but what I really want to highlight is that we can find these variants that are present in only a subset of ancestries using this type of approach. And these were prioritized with a posterior probability of inclusion greater than 90%. So summaries: to use FlashTree multi-trait find mapping, you do need the variant measured for all traits, and it does allow missing trait measurements. You can get improved resolution when the traits share causal variants. If there is no sharing, you'll just get back similar precision to single trait find mapping, so no loss. MG/FM has all of the same features as FlashFM, but in addition to that, it allows variants to differ across ancestries and groups because you account for that. FlashFM 0 is a way for high dimensional multi-trait fine mapping using latent factors, and there's different advantages to doing that. And I think software to highlight there. Each method has its own website and examples in the software, and hopefully it's easy to follow if anyone wishes to use these. And of course, acknowledgements for everyone who's been involved in different aspects of the different work. And I will stop there for questions from everybody, for all of us. Thank you.
Super, Jen. Thank you very much. It's really interesting to see your work there and the fantastic new software, and also to get that example from outside the field of psychiatry and just be able to see how things work with more power in larger numbers, which I'm sure we'll get to. So I'm aware that it is on the hour now, and many people may have to go. Thank you for joining us. If you do need to leave, if you have a few minutes to stay on for questions, I think our speakers are all able to stay on. So please do so. You can add your questions in the chat. Johnny, maybe I'll take one of those for you, and then we can have time to move on to other ones. Perhaps you want to pick up on the first one, which is the first question. Sorry, I appreciate that. I think it's only us on the panel that can see it. But a comment from someone that about concerns about the word causal, which we use throughout genetics and very much in fine mapping, because this is often interpreted as genetic determinism with the public. And so is that a concern, and how could we do that better? Johnny? Well, any of
I mean, I don't know if this was Howard Edenberg's question, but it's certainly a question that Howard has raised in conversations in the past, and I think it's a really important one. I think it gets really at the an important question that what we have in complex traits is not like Mendelian traits. It's not like, for example, the expansion mutation in Huntington's disease, where you have something that is truly causal, that if you have the variant, you will develop the disorder. We obviously are talking about complex traits, which are probabilistic and about changes in risk. From a statistical perspective, I think these variants are causal of the signal. That's really what we're getting at. But I think there's a need to communicate that nuance clearly and succinctly. We've used the phrase contributing to causality in the past. I'm not totally convinced that really gets the meaning across. But I think it's something we need to kind of think about. Perhaps we should qualify our meaning with statistically causal. I think there's also, I am yet to think of a scenario in which you could have a statistically causal variant that isn't at least proximally biologically causal. It's not doing something biological within the region. So I think you could have an argument of saying statistically causal, causal of biological effects, but perhaps we do need to qualify that it's not causal of the phenotype itself on its own, not sufficient for causality.
Thank you. That's helpful. Jen, Maria, do you agree? Do you have comments or suggestions of another word or phrase that? No,
I agree with Johnny. We can use like either that or likely causal, but yeah, it's purely from the statistical
perspective. We're still using causal, aren't we? And
yes.
I don't want to speak for Howard, but I wonder if there is a slightly more general term that we could come up with that we can revisit.
One thing I'll say there is maybe just causal association is one way that we've tried to steer it a little bit. So I'm just saying causal variance, it does sound imply biological. So just say causal associations, and you're kind of going back to that it's coming from the GWAS summary statistics.
Yeah. And I know Carolina wants to ask a question, and anyone else that wants to ask one, please do add to the Q&A. Carolina?
First of all, thanks to everyone. It was really, really interesting, great talks. My question is, in the complex trait, complex disease field, it's clearly much harder to pin down causal variants, causal associations and genes involved in those outcomes compared to, high impact rare variants. And then a lot of the downstream follow-up seems to focus more on the latter, which is a problem because some outcomes like major depression don't really have a lot of high impact rare variant findings. So all of your work is clearly really helping, but where's the point where the field accepts that this has now been fine mapped? Here we know the gene, we're done with it, and people are happy to move on to functional follow-up or organoids or, you know, new drugs.
Yeah. Right. Good question. Who would like to come in on that? Johnny? Yeah.
I think it's a really good question. I don't necessarily have an answer to it. I think one of the challenges is that limitation that I talked about of what we are talking about most of the time is the best variant of the set we're looking at. And before we have large scale meaningful sequencing, which is not something that, as much as I think we would love to have, we're going to have in the short term, it's very difficult to sort of say, yes, we're that confident. I think there are variants where the weight of evidence says something is interesting. So the Furin-Fez one that I've highlighted comes up consistently across depression and bipolar and schizophrenia, and it seems like we could be reasonably confident that there's a level of triangulation there that's going on. I think there's also a level of triangulation from different methods. That's one of the things that I think Maria's work in bipolar is really nice, is bringing together a lot of different things to give you some elements of things to look at. I think also a lot of what the functional genomics group is hoping to do will have benefits there as well, of trying to bring together disparate evidence from functional information as well. So I think there's probably a layer of broad scale functional information that we can bring in to inform this statistical fine mapping as well, that will lead us to something that is a bit closer to then being able to go, okay, let's go and do the expensive work in mice and zebrafish and so on. Yeah.
So Johnny, you're saying that's a question of judgment rather than a specific sort of threshold or piece of information? No?
I think so. I think until we're at the point where we can go, here's a bunch of sequencing and we're confident that this is the statistically the variant, I don't think we can from that perspective say it. I think it has to kind of come from the functional side. But it's also a question of like, is that what we're actually necessarily trying to achieve here? You know, functional information, fine mapping information can be useful from biology, but it could also be useful for prediction as well. It could improve polygenic scores.
And Howard has also put a comment in the chat that this is a graded response and prioritizing based on initial analyses with follow-ups, investing more as our understanding increases. And that's sort of.
And just one more comment to
Maria.
Just one more comment to this one. I think this will be done sooner rather than later for regions that have less than 5 or 6 genes, you know. Like when you get to regions, you know, like more than 10 genes, like the broader regions, it's even harder to do that. But I think, you know, like doing that, then the different layers of evidence can help us, you know, disentangle this landscape.
Mm-hmm. Yeah. Mm-hmm. Thank you. And Natasha's also commented on having one causal variant may not be actually what is the most meaningful for us there. And I assume, Natasha, you mean there in comparison to having a gene or that would take us to the next level. Right, I can see that it is 10 past the hour, so I think we should wrap up here. Thank you to all our fantastic speakers, Jennifer Asimit, Maria Koromina, and Johnny Coleman for their fantastic session. We've had a really high attendance at this one, and I'm sure that will be that it'll be very popular on the recording as well. Thank you to all of us for joining us. We'll finish here, and we look forward to seeing you at future talks, and also to say that we always welcome suggestions of topics to have sessions on or offers to speak. Please do email me if you'd like to be standing up here with your peers on the panel. We look forward to hearing from you. Thank you all. Goodbye.